All right. Pictures out. Hold them up. I saw it. It's beautiful. It's beautiful. Looks good. Yes, yours is very colorful, though Jaden's is too.
Mine's purple.
Yours is purple. Let's see. Where is it? Nice. Why do people not show up to class?
Anna missed the bus. She's walking from Monroe right now.
Well, good. No, I just wanted you guys to be familiar with it before we talk about it. That's all.
Mine's beautiful. How dare you?
I think the human body is ugly, but all right. So today we're going to talk about bacteriophages primarily, and the reason why I wanted to start here was because one, bacteriophages, good lord, are the... Alright, so bacteriophages are a great example of viruses because basically what we see here is somewhat analogous to what we find with eukaryotic viruses. So that's why we usually start here. The other reason why I wanted to start with this one was because it's a simpler model. We know more about bacteriophages than any other virus, or at least the general life cycle of a bacteriophage. And then also, it is a very good model. So the steps that we see today, or that we talk about today with infection, we'll see again when we talk about eukaryotic viruses as well. The last reason why I wanted to cover this is because bacteriophages have actually gained a lot of attention again in the past probably 10 years or so. So first discovery of bacteriophages goes back to the 1920s. Félix d'Herelle, who was a French scientist, had basically found these in part on accident, and his research led to using bacteriophages as a way to treat cholera infections within India in the 1920s. And around that time, there was a lot of thought that bacteriophages would become the primary way in which we treat bacterial infections. That was true until about the 1940s when penicillin became the most prominent drug used to treat infections. And for quite a long time, bacteriophages became more just a tool to study molecular biology than really to use to combat bacterial infections. Though that story is true in the Western world, bacteriophages have been used to treat illnesses, particularly skin infections, in Russia and other Eastern European countries since the 1940s. And within the past 10, 15 years, there's been a lot of renewed interest in using them for similar purposes in the United States. So using them in conjunction with antibiotics to treat infections, using them in place of antibiotics to treat infections, and then also some of the foods you may eat already, bacteriophages are being used to treat those foods. So for example, Listeria monocytogenes is considered a zero-sum food, or a zero-tolerance food. In other words, since Listeria is a psychrophile, can grow at refrigerated temperature, any food item that is refrigerated or frozen that may have Listeria monocytogenes, it has to have no detectable Listeria when it goes to the market. Okay? There have been a number of outbreaks in the past couple of years of times where with ice cream or with deli meats where Listeria infections have been quite prevalent. There's a company that sells a cocktail of 5 or 6 different bacteriophages. It's called Listex. Basically, it's kind of like a spray that they put over the top of the food as a way to treat it, kill Listeria. It's not disgusting, though. It's not disgusting. And the reason why it's not, it's just like putting a chemical on there, or a natural chemical, completely organic for those of you that are into the organic craze. All right? It's completely organic and does no harm to us. Bacteriophages do not infect human cells, and that's why it's not disgusting. So, if you think that's disgusting, just wait until other things in life. Anyway. All right. So what I want you guys to do is I want to start out with the following. You've all drawn structures of the bacteriophage. I want you to describe to me what is the purpose of that actual structure. So this here, what is the function of this? Okay? That whole thing, what's the function of it? I'll give you a minute to think about it, talk it over with somebody else.
Did you look at the homework?
Yeah, I'll look right now.
Why is it
I'm
not?
sorry you had to walk.
Tell him. He was like winging everyone.
I was across the street and 4 minutes early.
I was really long, but I was like, I feel like every time I heard like that, I'm supposed to make it look right, like
All right, what's the purpose of it? Yeah, broadly speaking. That's a better answer. What else? Hey guys, listen up. Honestly, guys, real quick side note, I'm sorry, but you gotta stop talking. And this not only goes for in here, but this goes for John too. Both of us are trying to help you guys learn, and the amount of talking that's going on behind us lately is over the top. This is not high school, okay? So let's get a little bit serious about this. And this is not only important in here and important to me, but it's also important to your careers. Show some respect to the people that are trying to teach you some stuff. You're gonna have to show some respect to your patients too. So let's actually pay attention in here, okay? Inject DNA is one. What else? What else does this structure serve? This is found only outside of the cell. Okay, never found inside of a bacterial cell. One other reason, one of the things that I didn't draw in here, the DNA. This is a vehicle not only to inject the DNA and to deliver DNA from one cell to another, it's also there to protect it. Okay? So outside of the cell, DNA can be easily destroyed by nucleases and other enzymes that can break it down. The protein protects it from being destroyed. Okay, so those are the two purposes of this basic structure. When this comes into contact with a bacterial cell, how does it actually attach? How does that attachment occur? Yeah, with those things, right? The legs. So the tail fibers are involved in the attachment, and what it can attach to are a number of different types of receptors. It can be an outer membrane protein, could be lipopolysaccharide, could be flagella, number of different types of proteins and non-protein structures that it could attach to. At this point then, and there's been some really cool studies done on this recently in looking at how does that bacteriophage change when it comes into contact with the bacterial cell. One of the things that's been noticed is those tails almost kind of like bend or break once this phage binds to the cell. And in doing so, that bend that occurs, it takes that base plate and basically pushes it into the outer membrane of a gram-negative or into the peptidoglycan of a gram-positive. What's on the base plate here? This bottom portion, what's on there? So there's little spikes, but what do? What are those spikes made of? What was that? Yeah, it's the lysozyme. Anybody remember what lysozyme does? Breaks down the cell wall. It hydrolyzes the cell wall. So in doing that, when this base plate gets pushed down, it breaks apart or starts to hydrolyze that cell wall so that at this point what we get is a little, it's basically almost kind of like an injector, protrudes from the tail, and it serves as a conduit now for the DNA to get into the cell. And so in this case, bacteriophage, unlike eukaryotic viruses, The only thing that actually makes it in is that piece of DNA. At that point, that protein structure doesn't matter what happens to it, really. You oftentimes becomes degraded shortly afterwards, but it's served its purpose. Okay. We'll come back to lysozyme again in a few minutes towards the end of this. All right. So what I wanted to do with the time that we've got then is to kind of walk through the basic steps here, and this is why the worksheet that I handed out is like the one that that I put up on the slides for you. So I want to walk through the steps of what are the general basic parts of an infection, from attachment to exit of the viral cell. We've got, in the case of bacteriophage, we've just addressed actually the first couple of steps. We've talked about the attachment of the virus to the protein on the outer surface. We've talked a bit about penetration, or this, what also might be called entry in eukaryotic viruses, where the DNA or genomic material is actually making its way into the cell. The other thing I want to point out here, just like with humans, there are certain viruses that are specific to humans. There are certain viruses that are specific to dogs, cats, turtles, fish, etc. Though the same is true in the bacterial world as well. A bacteriophage doesn't infect all bacteria. It's oftentimes very specific to a singular species, maybe even sometimes specific to a certain strain or a set of strains of species. And I want you to kind of keep that in mind, not so much for today, but for the next couple of days as we begin to go back to discussing antibiotics again. Last three steps here: biosynthesis, assembly, and then release. And we're going to take a look at this and correlate it with this drawing here then. Yeah, don't need to write down all of this, okay? 2 stages to the viral life cycle. Not all bacteriophages go through both of these stages. All of them do go through what we call the lytic cycle. And this is, when we think of infections in our own body, this is how most of those viral infections occur. We call it a lytic cycle. Virus gets in, causes symptoms, gets out within 24, 48, 72 hours, etc. And we may feel ill a little bit longer after that as our body is recovering. In the bacteriophage world, this lytic life cycle lasts maybe 40 minutes to an hour in a fast replicating bacterium like E. coli. And what you can observe is from the initial infection to the point of release, you can see viral numbers go up to about, if you start with 1, can end with about 100. So you get 100 new viral particles for every 1 that goes in. In laboratory conditions, so the stuff that myself and then a couple of students that work with me, we can probably overnight from a culture of E. coli grow and get about phage on the order of about 10 to the 12th per milliliter, whereas normally with E. coli it's around 10 to the 9th, 10 to the 10th. Okay, so about 100 viral particles for each cell. On occasion, we will find this lysogenic life cycle, and we'll come back to this in a couple of minutes. But this top part here is really pointing out both the attachment and the entry or the penetration step. So the viral particle attaches to the cell, and then we get penetration of that DNA into the cell. Now, the DNA is the only thing that enters. So how do we get more virus? How does that occur? Obviously, we've got to have replication, transcription, and translation, but how does that happen here?
Different depending on the type of DNA, or the type of genetic material.
Yes, possibly the type of genetic material does have a control on how this will occur. And we'll spend more time with that on Monday, because we're going to talk about HPV, which is a DNA virus. We'll talk about HIV, which is a reverse transcriber. We'll talk about what that means. And then influenza is an RNA virus. So it can be different on the genetic material. bacteriophage, 24 out of every 25 of them, 96% are DNA viruses. So let's assume that this is a DNA virus. If only that genome is entering, how do we get more? Comes down to it uses the host. And that's true for every single virus. The host becomes a factory for producing more viral particles. So viruses can't replicate on their own. They have to have a host in order for that replication to occur. In the case of bacteriophages, that means in replicating its DNA, what does it have to use? What from the host will it use to replicate DNA? This goes back to maybe a week ago. What was that? Ribosome? Not for, it will use the ribosome, but not for replication. DNA polymerase for replication, ribosome for translation. What would it use for RNA synthesis? Or RNA transcription? RNA polymerase, yeah, exactly. So this virus, at least in the initial stages, is dependent on RNA polymerase and the ribosome from the host in order to make proteins. And once those proteins get made, then these bacteriophages oftentimes have their own DNA polymerases as well, that once everything is made, now they can start replicating really fast. Genome size, small. 100,000 base pairs is the typical size, about 2% the size of a bacterial genome. We know that some bacterial cells can replicate about once every 20 minutes. And they can replicate 5 million base pairs in 20 minutes. What's 20 divided by 2%? Less than a minute, like a half a minute maybe, roughly, to replicate a viral genome then. That's about the rate at which these are working. So incredibly fast to replicate, incredibly fast transcription and translation. And the assembly of these, it's completely random. So there's some great videos online looking at how this random motion actually leads to the formation of the viral structure. But you can kind of think of it as like if you were able to some way shake a bunch of puzzle pieces and then those puzzle pieces just self-assembled to give you a 1,000-piece puzzle, the actual image. That's what happens here, is that through random motion within the cell, we get attachment of the various components of these viral particles together to reform that viral particle, that each one with an individual genome that exits the cell. This is almost perfect, almost all the time. But we talked about on Wednesday, we talked about a form of horizontal gene transfer that involves bacteriophage. Anybody recall which one that was? Bacteriophage moves genetic information from one cell to another. Not transformation, transduction. Yeah, the other one. And what we find is that this step right here in the lytic life cycle, this is where the transduction events are likely to occur. One of the things that bacteriophage do in their assembly is, and after biosynthesis, but before and during assembly, what we will find is that they actually chop up the bacterial genome into small fragments. And that's a way to stop replication of the bacterial genomes and ensure that everything is occurring for the purpose of the virus. In those cases in which those genomes get cut up, on occasion we can get cases where these viral particles self-assemble around a piece of a bacterial chromosome. And that's the transduction event. And this is, again, this is a rare event. We're talking about maybe once out of every 100 million viral particles that get produced, this event takes place. So it's pretty darn rare. Most of the time, productive viral particles get produced. Last step, exit, or the egress. This one's showing these kind of blowing out the side, and this is really what happens. Let's look ahead a couple of pictures real quick. So bottom right-hand side there, that's a bacterial cell, E. coli. Here is the remainder of that peptidoglycan, was a bacillus cell, and basically all these white particles in there, each one of those is a virus. Like, this is the stuff that nightmares are made of, okay? There was a book that I used to read. I can't remember what it was, but it was some like scary story book where there was one about like somebody that got a scab on their face and then it opened up and a bunch of spiders came out. That's what this reminds me of, roughly. Okay. Just a quick explosion in the number of bacteriophage. We immediately can go from detecting 1 per cell to 100 per cell within a matter of minutes here. Now for this rupture to occur, I mean, this looks like things that we've talked about before. Other cases in which lysis of the cell has occurred. So for example, what kind of enzymes might be driving this process then? We've already brought it up once today. What's that?
Lysozyme?
Yeah, lysozyme, or what's known, the more general term that gets used, lysozyme is an example of it, are lysins. L-Y-S-I-N. So the purpose of these lysins is to weaken that cell wall. And as this biosynthesis is occurring, lysins are being produced, weakening that cell wall, so that eventually what happens is the cell ruptures, the virus gets out. Some eukaryotic cells, or eukaryotic viruses, have a similar mechanism of just rupturing or killing the cell. Most often, though, the mechanism is a little bit different, where that cell remains alive for a short period of time and then just dies because it's been so damaged. So that covers lytic. Majority of the time, lytic replication occurs. I'm realizing I forgot to do something, so we're going to have to come back to something a little bit later. Just side note. Lysogenic, oh, go ahead, Zach.
So is that the only way a virus can get like out of your body? Is that why they say you just have to wait out viral like infections? Or is there something about the structure that doesn't allow medication to?
Yeah, that's a great question. So Kathleen will talk about this more on Monday, but it doesn't hurt to start covering it now. There is a reason why we can't treat most viral infections, and it's because we've already said it, at least in the case of the bacteriophage here, the host cell becomes a factory. And so in order to block the replication of the virus, you have to block the host cell in most cases. In other words, any effective medication would have to be one that, or at least, how do I want to describe this? We would want to treat with medication that, just like with antibiotics, is selectively toxic. Toxic to the virus, but not toxic to our own cells. But that means then all viruses, all human viruses, use our ribosome. So you can't block protein translation. That's out of the question. Because if you block the viral translation, you block translation in our own cells as well. So what makes treating antivirals so challenging is that there's a limited number of targets. Most human viruses, gosh, it ranges from like, some have only 5 genes, others have 100 genes or so. And trying to create a tailored drug for each one of those has really been challenging. Now we do have some exceptions. Like Tamiflu has been used in cases, and I think is given prophylactically during flu season in some hospitals and nursing homes to the workers so that they don't spread flu to other individuals. And that's because we have that drug specifically blocks the ability of the virus to attach to a cell. So we've come up with some ways. For viruses, honestly, the best way that we have found, preventative rather than treatment during. Preventative in the form of vaccines is much more effective. Typically. But that's a really good question. So the tough part is it's just challenging to find something selectively toxic. Yeah. Lysogenic life cycle is much more rare. This is going to occur only in some bacteriophages. They have this lysogenic life cycle, and for maybe every 10,000 infections you might of lysogenic bacteriophage, you might get one that actually goes into that particular life cycle. In this case, after that plasmid is entered into the cell, or after that genome is entered into the cell, it integrates itself into the host genome. What's different between this and transduction is a couple of things. One, it's a viral genome that's being integrated, not a bacterial genome. And then the second thing that's different is this doesn't require any sort of recombination or homology as transduction occurs. So in this case here, we get the viral genome integrated. Sorry, I'm drawing my finger the wrong way, aren't I? No. Why does that go that way? That makes no sense whatsoever. That should be that way. I apologize. This is the first time I've ever seen that. Huh. Okay. Cell replicates. Each daughter cell would get a copy of the viral genome along with the host genome. And this just continues. The thing about lysogeny here is this is kind of like analogous to what we would describe as a latent infection in our cells. So if you've had chicken pox, which I'm guessing most people in here haven't because most of you have now had the vaccine for chicken pox, lucky you. If you have had mono, if you have had herpes of some sort, if you have had, nobody in here has probably had Kaposi sarcoma and known it. Those are the major ones, all right? So if you've had one of those two, those viruses go into a latent infection. In other words, the genome, if you've had EBV, if you've had mono, you still have the virus in you, and you will never get rid of it. And in most of us, it won't do anything ever again. It will just remain dormant most of the time. One of the things that we know, what is one thing for herpes virus, and I'm not talking sexually transmitted here, I'm just talking regular. What oftentimes associates with a flare-up of cold sores? Stress. And that's exactly what happens here. Some sort of stress triggers the integrated bacteriophage to exit from the cell and go right back into a lytic cycle. So there's a lot of analogy here between what we know about eukaryotic viruses and bacterial viruses, at least in this sense of lysogeny having some correlation with latent life cycles of human viruses. And really what happens is just some sort of stress. It might be in the form of UV radiation. It might UV. It might be in the form of some sort of other DNA damaging agent that triggers the virus out of this proviral or lysogenic life cycle and back into a lytic life cycle. It's as if whatever that stress is telling the virus, oh, it's time to get out of here, the cell may die. Essentially, that's kind of what the cue is, right? And this can be really beneficial. So one of the things to think about here is there's really not a downside to either of these life cycles. Maybe there's a bit, but there's an upswing to both of them. So what I want to do is for a couple of minutes, I'm going to put up a picture here. And this is one I didn't lecture on. So this is a picture showing kind of just overviewing what might happen in lytic versus lysogenic life cycle. So just to kind of review what we've seen so far, in lytic, virus gets in, replicates, assembles, gets out, 40 minutes. In lysogenic, virus gets in, integrates its genome, hangs around in the cells as they divide, some sort of environmental stress triggers them back in the lytic cycle, they get out. Thinking purely from evolutionary standpoint, right? The goal of any organism when it comes to evolution is pass along your genetic information to the next generation. Biologically speaking, I should be dead now because I have succeeded in that. I've passed along my genetic information to 2 children, all right? Repopulating the world. Not Duggar style, but 2, okay? How does that hold true here? Okay, so think about this through an evolutionary viewpoint and think about what is the advantage, or when might these 2 different life cycles, lytic versus lysogenic, when might these be evolutionarily advantageous to the organism? Okay, I'm going to give you a couple minutes to think about this. This is a little bit, this is in some ways a little bit more challenging than it might seem on face value, and we'll walk through it afterwards. But why? Why have these 2 distinct life cycles? Okay? All right, go ahead. I'll give you a couple minutes. Is if you think of this in terms of like feasting and famine. Okay? In our gastrointestinal tract, we have a lot of lytic bacteriophage. Benefit being, bacteria within the gastrointestinal tract, for example, Bacteroides, which we'll talk about next week, somewhere on the order of about, what's 10 to the 11th? 100 billion, roughly? 100 billion per gram of feces. Yay, a lot, okay? That to the bacteria, or to the bacteriophage that infect Bacteroides is feasting. There's a lot of host to replicate in. Now imagine a different condition, something like a water condition, where bacterial populations are quite low. Is it advantageous to kill the one or two hosts that are available at any time? No, it's like a bottle rocket versus a real firework. Right? So it's popping. Oh, that was great. So what I'm trying to point out here is that in the case of lysogeny, the benefit to the organism is that its genetic information can continue to be passed without the virus having to do any sort of work whatsoever. And in times when there aren't many hosts around, this can be advantageous to ensure that that genetic information continues to be passed. This method here also allows for a wider dissemination in some ways. What do I mean by that? So, what would be a good example to this? HIV is probably the best example that we have in comparison. Somebody's infected with HIV, they're infected for the rest of their lives, right? But on the outside, for most of us, you wouldn't know, at least these days with the antivirals that we have, you wouldn't know if somebody was infected with HIV, right? And that allows for people to move around without being detected as being infected. Same thing is true here. As I walk around this room, touch, duck, 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 I have now spread bacteria from this computer to this, and I'm now going to drink out of that later, and then over here as well. And then I'm going to go home this evening, and I'm probably going to eat dinner, and there's going to be bacteria all over the fork that I used to eat dinner, etc. Right? That's dissemination. That's spreading the bacteria from one location to another without any of them really knowing that they're infected in some way. And we see that with human populations as well. As we have international travel from one location to the next without people knowing that they're sick, it allows for us to disseminate viruses to new locations, introducing that virus to brand new hosts that weren't available beforehand. Okay? That's the benefit of lysogeny, is allowing the virus to move into a new area, covertly, but into a new area, going undetected, so that now it has possibly the ability to infect more hosts than it originally had. Does that make some sense? Danica?
But even though it's like moving to a new location, it won't necessarily be like activated, or like won't necessarily.
Not necessarily. You're absolutely right. So the movement to a new location does not ensure that an activation out of lysogeny back in the lyticus cycle would occur. That comes from some sort of environmental trigger that, and oftentimes it's some sort of trigger from damaged DNA in the host cell. Yeah, that's a great question. Sarah, did you have one? Okay. All right. Let's see if there's anything else in here. I think that was all I wanted to cover today with this. So, any questions? Again, this is kind of a broad overview. The main takeaway points today, at least, are just thinking about how that structure of the virus fits the function of it. And then I want you to just have a basic idea of the lytic and lysogenic life cycle and be able to compare the two with each other. Right? Those are the major points for today. One thing I completely forgot to do when we got in here today, I wanted to go over that question we finished up with on Wednesday and talk about transformation for a couple of minutes. Yes, it was the really bad mouse drawing. Really bad mouse drawing.
The first one was good. Your second mouse looked like he died.
I tried drawing them today, and they were really bad. All right. So to remind you of this problem.